So in lab this week, you guys did muscular and skin and integument and so on, and looking at muscles and joints and movement. And this coming a week after Thanksgiving, you'll be looking at excretion, circulatory system, and so on. So the kinds of things we're talking about right now are going to fit perfectly with what you guys are doing in lab. How's the volume in the back? A little loud, or is it okay? It's okay? Sounds loud to me here. Okay, I wanted to remind you of a few things from last time that are going to lead in really nicely to what we're talking about mostly today, which is internal transport. So we're going to be looking at how do animals move materials around inside their bodies. That's partly gas exchange, getting oxygen in, carbon dioxide out, partly nutrients, includes things like hormones and all of that. That's related to some things we were just finishing up on. So last time, very quickly, I went through 4 ways to gain or lose heat. In this case, I showed animals gaining. But those 4 ways, radiation being the first one, each one depends upon the temperatures around you. So radiation, you're radiating away heat. The surface of anything radiates out in proportion to the surface temperature. And it gains back according to the surface temperature around the outside. One of the cool things about this is that it helped me understand why, when I was a kid in Iowa, in the summer, it could be 72 degrees in our poorly insulated family room, and I would be really hot. And in the winter, it could be 72 degrees, and I would be freezing, which never made any sense to me until I realized that in that poorly insulated room, the walls and roof were radiating different amounts of heat. In the summer, even though the air temperature was 72, the walls were really hot, could be in the 90s. And so they were radiating in a lot of heat toward me, and I was gaining a lot more heat from the walls. In the winter, the walls might be 50. So even though the air temperature was 72, much less heat was radiating into me than I was radiating out to the walls. So the temperature I perceived was really different, even though the air temperatures were the same. Chloe?
How does the radiation not also heat up the air?
So how does radiation also not heat up the air temperature? Because oxygen and nitrogen just allow that radiation to pass through without absorbing it. Now, carbon dioxide and methane, so if we filled that room with carbon dioxide and methane, those would pick up that radiation very, very nicely. This is the problem with greenhouse gases. So it's exactly the deal with greenhouse gases. However, carbon dioxide, 0.02% of air. Hopefully your house, and mine too, does not have much methane in it. It would be a bad thing, potentially explosive family room, not just because of family dynamics. Again, these jokes are for me, not for you. Okay, then conduction, whatever you're physically in contact with, which could be solids. It also can be still air. Both of those are conduction. If you have warmth, in other words, a lot of fast random molecular motion on the surface of your skin, you are transmitting that. You are running into, bouncing into molecules on what you're sitting on your clothes, and in the air. That's conduction. Convection is sort of the same thing, except we're speeding it up. With convection, we're sending a breeze or a current past something. And so when the heat is transferred, it doesn't transfer back. So if you bring in cold air, you transfer heat to it, and then it gets carried away. It can't come back to you. Whereas if you're just sitting in still air, the molecules you heat up, they are just as likely to bounce back and hit you as they are to bounce out further away. Okay, so convection really speeds up heat transfer. And then evaporation. Of course, which molecules escape hydrogen bonding? It's only the very fastest ones, by definition, the hottest. And so if only the fastest water molecules can escape, then evaporation is going to leave behind the ones that aren't moving as fast. It's cooler. But of course, that only works for terrestrial animals. Evaporation is a meaningless concept for animals in water. And again, what can animals do in response to temperatures around them that are not at their favored? They can tolerate it, they can adjust to it, they can move, or they can die. So when we're talking about some of those adjustments, we have two ways to think about how animals do this, and they're two continua. In the one case, temperature changes with the environment versus body temperature is always the same. So temperature changes with the environment, or body temperature is always the same on the right. And those are, if the temperature changes with the environment, we have a terrible term, poikilotherm. Even sounds like an uncomfortable word. It's like the most uncomfortable chair in the house would be a poikilotherm. And a homeotherm. Homeotherm, body temperature stays the same all the time. We are homeotherms, although not perfectly. Right now, probably your hands are considerably cooler than your core. And we change a little bit. On a daily cycle, you change by about 0.5 degree centigrade. So we're not perfect homeotherms, but pretty good. In really hot weather, you'd increase your temperature outside by 1 degree or 2. Okay, so that's one continuum. The other continuum is for the source of heat in the body. Two possibilities are heat only from the environment. That's an ectotherm, versus generating one's own body heat, that's an endotherm. We are homeothermic endotherms. The two can be independent of each other. A lizard is a poikilothermic ectotherm. Body temperature changes with the environment, and body heat comes almost entirely from the environment. But there are homeothermic ectotherms. So think about a deep sea organism living at a constant temperature of maybe around 5 degrees centigrade. The deep sea is a constant somewhere around 5 degrees centigrade. And if you're a deep sea animal, you are a homeothermic ectotherm. In other words, you can have selection acting to maximize function of enzymes at 5 degrees, just as you have selection acting to maximize function of enzymes at about 37 degrees centigrade. Okay, does that make sense? Okay. We're going to talk more about countercurrent heat exchangers when we talk about gas exchange, but I want to mention a couple things. When you're talking about a countercurrent heat exchangers, they can seem pretty simple on the surface, and yet on the exams or when people try to describe them to me later, they get them backwards. They get them messed up. So I'm going to give you a checklist every time you think about a countercurrent heat exchanger. It's really helpful to identify the source, in this case of heat. We have hot blood coming from the heart. And if this was a wolf standing on ice, or a seagull standing with its feet in freezing cold water, or a heron, the source is heat, hot blood from the heart. And there's a sink, cold blood from the skin or the foot. And of course, in this case, we've got a cycle. Going down to the foot. Okay, so the heat is going to go from the source to the sink. So the checklist is there only one source and is there only one sink, and have you identified them properly? The next part is, are the source and the sink at opposite ends? Okay, and we have them at opposite ends. The sink is coming in low in something, and it leaves with the transferred whatever it is to it. The source is coming in high in something, in this case heat, and at the other end it leaves with much less. So we've met that condition, and is the transfer always from the source to the sink, always from the higher amount to the lower amount? Okay, so let's look at that. We have hot blood from the heart at 37 degrees, and the blood coming back down below is cooler. Does that make sense? It's always coming from the left side here, and it's always going to be cooler. And so we should have heat transferred in that direction, from the warmer to the cooler area. Make sense? Okay, so as we go along from the source, we have cooled down that blood. It's now 30, but it's encountering blood down below that's cooler. So again, we have heat transfer. Further, again, 20 to 18, 10 to 8, and we can keep going until we were down to something just barely above freezing, which is what happens in a heron standing in cold water, or a seagull, or a wolf standing on ice. Now, it would happen to you too if you were standing barefoot on ice, but your feet don't tolerate that well. You cut down the blood flow to your feet, so even if your feet don't freeze, that cold closes off capillaries in your feet, and you get what's called frostbite. You basically are oxygen and nutrient depriving your feet. Okay? And if that goes on for too long, you get cell damage. Cells start to die. But a wolf, or a heron, or a seagull is adapted to tolerate those low temperatures. They do not get frostbite under the same conditions. Okay? We are semi-tropical animals, tropical to semi-tropical. We are not well adapted without exterior coverings for really cold climates. Okay? We are still fundamentally much more tropical than we are temperate zone animals, and certainly not Arctic. Okay? Okay, so is transfer always from the source to the sink, always from the higher amount to the lower amount? Keep in mind, the numbers won't necessarily tell you, because if this was oxygen, well, I guess it'd still be higher amounts, so higher amount of heat. But do think about what is it you're transferring. Okay, don't just memorize the numbers, because it's really easy to get things backwards. Okay? Okay, take a moment, explain this to your neighbor really quickly. See if you can picture it with your eyes closed too, if you want. Okay, a few more concepts before we start talking about circulation. Metabolic rate is the amount of energy an animal uses per unit time. That's directly related to oxygen use for aerobic animals. And we are selected to have high metabolic rates. Those high metabolic rates give us faster movement, faster processing, which is really important. With a big complex brain, we are slower. Many more connections. That slows down what we do. Without our warmer body temperature, we'd be really slow to respond to any complex stimuli. So we have a high metabolic rate, high energy use per unit time. That puts constraints or requirements on our circulatory systems, our gas exchange systems. You have a very high pressure circulatory system. You have to. With our high metabolic rate, if you did not have a high pressure circulatory system, high pressures out to your systemic circuit, you would be starved of oxygen and glucose, and you'd be overwhelmed with the amount of carbon dioxide you produce. Okay? You have to have that high pressure circulatory system. We talk a little bit about metabolic rate under different conditions. It won't matter that much to us, but basal metabolic rate, when we want to compare animals, that's the metabolic rate when an animal's just sitting doing nothing special. In what's called its thermoneutral zone, so at a temperature at which it does not have to exert any energy to either warm up or cool down. So if you're too hot and you have to fan yourself, you're not in your thermoneutral zone. You actually have to generate heat and use energy to cool yourself off. Okay. If you're cold and shivering, again, you're generating heat to warm up. If you're in your thermoneutral zone, you're doing none of that. And if you have a sweater or whatever on right now and your temperature is comfortable, you're in your thermoneutral zone. Okay, that would be basal metabolic rate. You're a little bit above that right now, but not by that much. Some animals drop their temperatures for periods of time in things like hibernation. So mammals, for example, often change the set point of body temperature in their hypothalamus and drop down, for example, ground squirrels in winter, burrowing down into the soil, being at a depth where their surroundings are quite cool, dropping their body temperature to down around 4 or 5 degrees centigrade. At that temperature, they're using somewhere in the neighborhood of 1/100th the energy per day. So the amount of food it might take them to survive a day at normal body temperature will take them through months. Now, it turns out they need more energy than that because animals don't stay hibernated through an entire winter. They periodically arouse, do things like drink, but they have to warm up. And so they need the equivalent of, often it's a couple weeks worth of fat to make it through a winter, okay? But it's not huge amounts to make it through. So that's one way in which even a homeothermic endotherm, us, may change temperature. And there are varieties that are not just hibernation. So some animals, some shrews, for example, will drop body temperature by about 10 degrees centigrade during the inactive period, during the day, because they don't actually have enough energy to make it through the full day if they stay at a high body temperature. The consequence is they would be slower in response to a snake or a predator or whatever. Hummingbirds have to do the same thing at night. Okay. And then the last thing before I take your question, Jess, the last thing is we also do fever. So you can change, so inputs to the neurons, the neural circuits that are controlling your body temperature, that are making you decide to put on a sweater or making you shiver. The number of action potentials coming in can change the set point, both to make it cooler, hibernation or torpor, or to make it hotter, fever. And adaptive fevers are fevers that don't risk damage to your nervous system, but they raise the temperature of your system enough that bacteria don't function quite as well. Invading bacteria don't function quite as well, and your own immune system's metabolic rate is slightly faster. So it improves your chances of getting rid of those invaders. Okay. The invaders, the bacteria, are selected to be able to invade you at your body temperature, raise the temperature a bit, and that's no longer their optimum. And so you have a better chance to fight them off. Okay, Jessica? So why don't humans hibernate or lower their body temperature? Larger animals tend to not hibernate as often. Few do, bears do, but oftentimes they don't. And that starts getting into more complex ecological questions that I'll happily talk with you about after class, but probably shouldn't talk about now. It has to do mainly with the relationship between how much food is available, what benefits the animal might get in terms of fitness for staying aroused. For example, if we hibernated during the winter, you can just forget about raising kids during that time. It slows everything down. Okay? All right, so any other questions? I could tell I was going to go off on a long tangent until I stopped myself. Okay, let's move on to talk more directly about internal transport. Now, you've watched some videos on this, so I'm just going to do some quick reminders at the beginning. So what is a circulatory system? Well, in us, it's this very complex system centered on a heart. And then blood vessels. And inside it, a circulatory fluid, which could be blood, and that's for what kind of circulatory system? Blood in an closed, okay, or hemolymph in an Okay, hemolymph in an open circulatory system. If we're talking about simple animals, if they're small enough, they don't need a circulatory system. So flatworms just don't have a circulatory system. The reason they are flat is that constraint. Diffusion distances for gases, effective diffusion distances for gases in water are about 0.5 millimeter. Okay, so somewhere around half a millimeter, maybe a millimeter. And we're talking about diffusion fast enough to keep cells alive. Okay. Of course, it'll diffuse if you give it long enough. But to keep cells alive, around 0.5 millimeter. So the reason flatworms are flat is without a circulatory system, that's all they can do. They have to have every cell within 0.5 millimeter or so of the surface. Okay, otherwise it's going to die. And then there are some animals that are simply thin. So corals, jellyfish, even though they may have a thick bell in the case of a jellyfish, they just have a single tissue layer or a few tissues around the outside. And then in the inside of a coral or an anemone, they have a gastrovascular cavity that they can move water in and out of, but it's just thin layers of tissue all around. So again, they don't need an internal circulatory system because they can move water past it to absorb oxygen and get rid of carbon dioxide. As animals get bigger, that no longer works. And so now we have circulatory systems. Here's a crayfish or a lobster, depending on which you prefer. There's a heart. And we've got blood going out. Okay, going out through the tissues. It is an open circulatory system. So there's no capillaries. Instead, the blood vessels, or the hemolymph rather, open, the fluid moves in between cells. And winds up going into a venule. So I've shown these red arrows here. It goes between cells. It's losing oxygen. It's picking up carbon dioxide. It's losing nutrients. It's picking up waste products. And it's going into vessels, going through the gills. And then back to the heart. In the case of a crayfish, some of it is just coming in through little openings, ostia, into the heart, and then being pumped out again. We have a tendency, let me make a comment here again, open, there's no capillaries in the gills either. Okay, we have a tendency to think, well, ours is a closed circulatory system, that must be better. In fact, there are advantages and disadvantages to both systems. The advantages of a closed circulatory system, the big one is you can make it faster. You can have high pressures. Second big one, you can easily change delivery to muscles, brain, kidney, whatever. You can really easily adjust, even on a local scale, even down in little patches of tissue, because you can open and close individual capillaries. So you can adjust how much blood goes through. You can also separate specialized transport molecules or cells in the blood from other molecules and cells. The only reason you can have red blood cells is because you have a closed circulatory system. Animals with hemolymph can't do that because a big cell would get trapped trying to go in between other cells in an open circulatory system. You can pack all of your hemoglobin into a red blood cell, and it doesn't get stuck anywhere because it's always staying within vessels where it fits. So those are the advantages of a closed circulatory system. But there are disadvantages. The big one is all that tissue. You have, I think in your body, the estimate is something like 50,000 miles of capillaries, or maybe it's only kilometers. Still a lot. Okay. That is a vast amount of tissue that you have to have, that an animal with an open circulatory system doesn't have to carry around, doesn't have to make, doesn't have to carry around. So you're putting a lot of resources into capillaries. The second is that for a closed circulatory system, you have to push things through it under pressure. And of course, you need gas exchange and other kinds of exchange. And so the capillaries need to be a bit leaky, which means fluid gets out. Where's it going to go? How are you going to get it back? So you need to invest, animals with a closed circulatory system like us, especially a high pressure one, we need a way to get that fluid back in. You have an entire additional system, the lymphatic system, with its own lymph capillaries and veins, starting as little capillaries in your tissues and coming back and draining into a major blood vessel near your heart. Whole additional system. So a closed circulatory system is expensive. And animals have all sorts of versions. The crayfish I just showed you doesn't have a lot of veins leading back to the heart. In fact, some of it just goes directly into little ostia into the heart. Whereas octopus and squid have really small arterioles going to every part of the body and really tiny venules picking that blood up. And so they have something not too different from yours that is a compromise. It gets many of the advantages of an open circulatory system without some of the disadvantages of a closed one. But nonetheless, they're still not getting as good local control of where the blood goes as you are. So that's what you're getting from your extra investment. Okay, so with a closed circulatory system, you have capillaries in two regions. There's a gas exchange region, and most animals need this. There's a few animals that get away with no specialized gas exchange region, but not many. Earthworms, for example, they don't have lungs, they don't have gills, they just have a lot of capillaries right underneath their integument. And those are picking up oxygen that diffuses across their most moist outer membranes. So they don't have a gill or a lung. But in most animals, there's a gill or a lung. And then there's the system. What we talk about is the rest. So your systemic circuit. In a human, we actually talk about a systemic circuit, which is blood going everywhere except your lungs and your head. And we talk about separately the system that is, or the part of your blood vessels that are going to your brain. I'm just going to call it all the same thing, all system. Okay. So in a mammal, what we have is the equivalent of two hearts. So we have a divided heart, but it's really as if we put a second heart between the area of gas exchange and the area and the system. In other words, we have an area for gas exchange, and then to the heart, and then out to every other part of the body, and then back to the other half of the heart. So it's almost like you have two hearts glued together, the right side and the left side. Okay, make sense? To remind you about structure, so fish, teleost fish, or sharks, we've got blood coming in. And remember, any heart is just a muscular chamber with one-way valves. Muscular chamber with one-way valves. That will pump fluid. So sinus venosus is bringing blood back. The atrium, just like your atria, it's thin-walled. It just has to get blood to the ventricle. Its function is to collect blood and then quickly fill the ventricle. It's got a one-way valve. And back pressure pushes that closed. And it has another one-way valve. Again, back pressure would push it closed. So when the atrium contracts, which one closes, A or B? Okay, A. When the atrium contracts, we increase the pressure. It pushes on the valves at A to close. What happens at B when the atrium contracts? Okay, pushes it open. Okay. And then when the ventricle contracts, notice how much thicker. When the ventricle contracts, which one closes, B or C? Okay, B gets pushed closed, C gets pushed open. Okay, any circulatory system has to have some elasticity in the vessels. Blood, water, is not very compressible. And so when it gets pumped out in a bolus in one mass of blood, we have to expand wherever it's going to take that blood. So it has to be stretchy. Your aorta has to be stretchy. Okay, so with every heartbeat, your aorta has to stretch out to take that blood. But it needs to be stiff enough to have enough elastic recoil that after it stretches, it recoils back, which can't go backwards, right? That valve stops it from going back to the heart. So elastic recoil pushes that blood further down the system. So your whole arterial system, if we could watch, with each heartbeat, your aorta coming out of the heart, okay, coming out from the left ventricle, would expand, and then elastic recoil would make it shrink, and you would create a bulge just downstream, and a bulge. And that bulge goes all the way through your body as your pulse. You can measure it even down on your finger. Okay. As a pulse, the pulse is the expansion of those blood vessels, the increase in pressure from the expansion of those blood vessels. Okay, same structure in a teleost. Here's the mammalian heart, although I never find this a very useful one to study from because it's too complex. The structural details matter if you really get into heart function. Okay, but what I really want you to notice now is the thickness of the right versus the left. Okay, which one is thicker? Okay, left and why? More pressure. Okay, you have a divided ventricle to generate those higher pressures. Okay, do you guys know how to draw a heart quickly? Do I need to run through this? All right, we'll do it quickly. Okay, here's your first line. Then you need 3 lines like this. These are going to be your blood vessels. Then you need a line here and here. You need one-way valves. Let's put some vessels coming in. and blood is coming in through these. These are veins. Okay? Then we need lines here with one-way valves. We need one-way valves in these blood vessels. And optionally, a septum. Took me forever to keep straight which went where. But I finally just started telling myself the blood goes right to the lung, right side. And very confusingly, it's always as if the heart is lying in front of you. And so the right side is on our left. Okay? Okay, if blood is coming back from veins and getting into the atria, and the atria contract, what happens to the valves? Okay, the valves from the veins close, right? They get forced closed. The blood hitting them on contraction is going to force them closed. So as soon as the muscle starts to contract, we're pushing blood against those valves and they close. What about the atrioventricular valves? They have names, but we don't care about them. What about the atrioventricular valves? When the atria contract, what happens to those? Can you see them getting pushed open? Don't just memorize it. I want you seeing that as you contract them, you're putting pressure on the fluid and it's going to push, and the way the valves are arranged, they will push open. Okay? So when the atria contract, you push blood into the ventricles. When the ventricle contracts, what happens to the atrioventricular valves? They're going to close, and you push blood out into the system in the lung. Okay? Okay, I mentioned that the septum is optional. So let's look at what happens when we compare an amphibian, or many reptiles, not all reptiles. This means oxygenated blood, not blood from an ox. Now, you would think that you're going to get a tremendous amount of mixing. But in fact, you get relatively little. I've seen estimates of 10%. I've even seen estimates down as low as 2 or 3% mixing. So how can that be? How can you send deoxygenated blood preferentially to the lungs, but not to the system? The reason that happens is that there's higher pressure in the systemic blood, not by a lot. It's about It's about 1.5 to 2 times the pressure in the pulmonary circuit. But what that means is that the valves going to the system open later. Does that make sense? If we were putting pressure on something, the pulmonary circuit and the systemic circuit both have back pressure, right? The blood is in there, it's under some pressure, and so while the ventricles are filling, those valves are closed. As the ventricles start to contract, which one do they pop open first? The one that has lower pressure here, or the one that has higher pressure? Okay. They're going to pop open the one that has lower back pressure. And that is the pulmonary circuit. So we open that one first, and the deoxygenated blood, because of the shape of the heart, the deoxygenated blood is collected mostly near that opening. So it opens first, and the deoxygenated blood gets pushed up into it. As the pressure increases, as the ventricle keeps contracting, the valve to the system opens up. That's where the oxygenated blood primarily is, and that collects mostly oxygenated blood. So you get very little mixing. Okay, does that make sense? You do get some. There's some inefficiency here. Any blood you mix is going deoxygenated back out to the tissues, not helping you. Okay, so there's some cost to that. But here's the advantage. If you're a frog or a turtle, some predator comes along, and you dive down to the bottom of that pond, okay, do you want to be the frog that has to come up after 29 minutes because you're out of oxygen and you pop up to the surface? Or do you want to be the frog that gets to come up after 30 minutes when the oxygen-starved frog has just been eaten and the predator is now full? Take your choice. If you can save a little energy by not sending blood through your pulmonary circuit when there's no oxygen there to get, you can stay down underwater longer. Make sense? Okay. So when a frog is not breathing, it just constricts all the capillaries in its lungs, sends almost no blood to the lungs. Almost all the blood just keeps going back through the system. Oxygen level keeps going lower and lower, but they have a relatively low metabolic rate. So it works. And even an extra minute or two is potentially the difference between life and death. For us, we cannot do that. In order for our high metabolic rates, you have a metabolic rate 5 to 10 times faster than a frog. Okay, for us, you need about 5 to 7 times higher pressure. The pressures in your lungs, somewhere around in that circuit, get to somewhere around 25 millimeters of mercury. Okay, not all that high. The pressure in your systemic circuit coming out of your heart, around 150. By the time it gets to your arms, it's closer to 120 unless you're working hard. But coming out of your heart in your aorta, close to 150. Okay, something like 6 times higher. With that big a pressure difference, you couldn't keep your blood from mixing. It's just not possible. So with that big a pressure difference, you've got to have a septum. Otherwise, you can't maintain those high pressures to the system. Okay. If you want a sense of what would happen if you did, there's one animal that has been strongly selected to have very high pressures in its pulmonary circuit. Those are thoroughbred racehorses. Thoroughbred racehorses get pressures in their pulmonary circuit that are really high because they have been selected now for well over 100 generations, probably hundreds of generations, to be able to run flat out maximum speed for a minute or two. And that requires really rapid gas exchange. And so apparently, without us knowing it, selection has favored really high pressures. They have pressures that can get up to 100 or more in their pulmonary circuit. This is not a good thing, except during a race. For years, people have reported thoroughbred racehorses bleeding from the noses after a race. Maybe 10, 20% of them do that. Turns out probably every thoroughbred horse after a hard race has burst capillaries in its lungs from those really high pressures in the pulmonary circuit. Okay. Not a good thing. Fortunately for thoroughbred horses, they don't have to do a lot of races. They don't have to go full tilt all that often, but not a good thing. And I'm sorry, it's been a downer, especially for all of you who really love horses. By the way, this has only been known, I think I ran across a paper just a couple of years ago that talked about this. Okay, while I figure out where I am in my lecture, you can think about poor thoroughbred racehorses. Okay, I put a few gee whiz facts in there. Typical heart beats about a quarter cup per beat, about 0.07 liters, so a quarter of a cup. Typical cardiac output in liters per minute is about 5, so you're sending about a gallon. Think of a milk jug, gallon milk jug. You're sending about that much blood through your heart every minute. Your total blood volume is only about 3.5 liters. That means you're sending your blood all the way through your body every few minutes. Okay. Now, if you were a mouse, you would be doing it 10 or 20 times faster. It's a much shorter distance to travel. Does that make sense to you? So similar pressures, but it's a much shorter distance to travel. So a mouse is moving its blood around faster. Which animal do you think can replenish its oxygen faster when it's exercising hard? You or a mouse? Mouse. Mice have metabolic rates somewhere around between 5 and 10 times faster than yours per gram of tissue. Confusingly, we talk about metabolic rates in 2 ways. One is total, just how much heat are you generating per minute, or how much oxygen are you using per minute. We also talk about metabolic rates per gram of tissue. Per gram of tissue, adjusted for size, in other words. A mouse has a metabolic rate 5 or 10 times faster than you. In total, of course, yours is much higher. You're much bigger than a mouse. Okay, so one of the consequences is the bigger you are, the slower your metabolic rate. Because you are having to move materials faster to replenish oxygen, to get rid of carbon dioxide, to get glucose to the cells and all of that. So really small animals, things with the very highest metabolic rates are very tiny birds and very tiny mammals. One of the consequences of being bigger is slowing things down. Okay, if we had a metabolic rate the size of a mouse, we could think 10 times faster. This would be a nice thing. Although none of you seem enthusiastic about it, but still. Okay, structure of the blood vessels. So structure of the blood vessels, just as you might expect. There's an epithelial layer. And it is called the endothelium, and that's in every blood vessel, all the way down to capillaries. So here it is again. This is a vein, the top one. The second one here is an artery. So here it is again in the artery. Here it is in an arteriole, still a capillary endothelium, and here it is in a capillary. A capillary has only that layer. It's got the epithelial layer, endothelium, and basement membrane. So all around it is basement membrane. Capillaries are leaky. It's not shown here, but there are gaps in capillary endothelial cells. Gaps between them, even little pores like donut holes, so that stuff can go through them, not in contact with the cytoplasm. And then the basement membrane keeps larger things from crossing. So typical proteins, anything from that size on up, virus, bacteria can't cross. That's a good thing. Then surrounding it, not surprisingly, you have smooth muscle and elastins, and the outer coat has collagens to limit the maximum size. Okay, next time we're going to talk a little bit more about transport across these, and a little bit more about the structure of the vessels, and then we will move smoothly directly from that into gas transport, into breathing and transport, because they're really two sides of the same topic. Have a good Thanksgiving. Exams are down front if you haven't picked yours up. Otherwise, I'll put them outside my office.